one of the strangest fossils just got a whole lot stranger. Prototaxites was by far the largest of the early land organisms, dwarfing the local land plants, and it's been debated as to what it was for something like 150 years. The most recent good idea was that it was a fungus, but a new study looking at the detailed chemistry of these things has shown that a fungus it was not. Most likely now, it seems to have been an entirely separate group of multicellular life, of which we otherwise know nothing. Life on land really got going in the Silurian and Devonian, although there are some spores suggesting that land plants evolved sometime a little earlier in the Ordovician. But we only really see proper fossils of them from the Silurian onwards. Now, a lot of these early land plants and related organisms are pretty odd by current standards. I mean, they hadn't evolved leaves by this point, for example, but we can still see that structurally and evolutionarily they are plants. There's no problem with that. I have a video about Cooksonia, the earliest known land plant, if you want to get a bit of background into them and how they evolved. Among these early fossils, though, there is something that is very odd indeed. Prototaxites has been a mystery since the 1850s when it was first found. It formed trunk-like structures up to over 8 meters tall and 1 meter wide, at a time when the early plants were measured in centimeters, or millimeters sometimes. So we're looking at a really gigantic structure. It would have absolutely dwarfed everything else that was around then. By the late Devonian, when Prototaxites went extinct, there were some other land plants that had evolved into trees which had actually started to overtop these things. But for the Silurian and into the early Devonian, Prototaxites was a giant. It was also very odd in various ways, in that there don't seem to have been any leaves, any branches. It's just an isolated trunk. Sort of flares out at the bottom. There's a suggestion from cavities in the ground that it may have had a root system, but that's never preserved. And the structure of it is not like wood. It's a network of little tubes of various sizes, all sort of interwoven to create this structure that is unique, basically. And you can look at similarities in things like palm trees, for example, and tree ferns, which have sort of tubular structure within the trunk, but it's not the same thing. So, going back to the early days, it was named by Dawson in 1857 on the basis that it was a partly rotten yew tree, or relative, hence proto, meaning precursor of, and taxites, meaning named after Taxus, the yew tree. And this was very quickly dismissed by his peers at the time, but at the time that was what it was thought of. There was a reconstruction of it as basically a conifer, without really anything in the way of branches, but a conifer-like thing. Okay, this was rapidly challenged, and because of this tubular structure, was identified instead as likely a gigantic alga. A land-living upright alga would be quite a feat, but structurally it made more sense than something like a conifer. So this then held sway for a long time. And it was only really until about 2000-ish that it started to be seriously challenged. It was left to one side effectively as a strange, gigantic thing that people didn't worry too much about. Most people didn't. Few people did, obviously. But come 2001, it was reclassified by a guy called Huber, or Huber, he was American, not quite sure how to pronounce that in that context, as a fungus. This reclassification was based purely on the structure, with Huber showing that it could be interpreted as being composed of fungal hyphae. So there were various different types of tube within the structure. 2 large ones, one with thin walls that was septate and one with much thicker walls that wasn't septate. By septate, I mean subdivided inside it. So this is a feature that we see very commonly in, for example, fungal hyphae. Fungal cells tend to be septate. Then there's also the third type, which is much thinner, also septate, and which wove around the others and bound the whole thing together. They often branched and basically formed a massive structure which held everything together. And okay, you can't really find anything in the modern world of fungi that looks particularly similar, but on the fundamentals of structure, it's not too far off. So he described this, or reclassified it, as a fungus, and in particular a basidiomycete, based on comparisons with some familiar types of mushrooms, like the milkcaps. Okay, so this, of course, is a totally different kettle of fish from an alga or a land plant, because they live in a fundamentally different way. So plants and algae are autotrophs. They get their energy and their food supply, they fix the carbon, from the atmosphere using sunlight. Fungi are heterotrophs. They get their carbon by breaking down other organic matter. And this gives us a way to actually test whether it is one or the other. Because when you get your food sources from these different places, it leaves a different isotopic signature on the carbon within the tissues. So when carbon is processed to become part of an organism, it undergoes fractionation. So the lighter and the heavier isotopes of carbon are used in different ways. In respiration, you tend to get rid of the lighter isotope and concentrate the heavier one. So you end up with a different balance of the isotopic ratios for heterotrophs versus autotrophs. In 2007, there was another paper by Boyce et al. In geology, which actually studied the isotopic compositions of Prototaxites, and found that they were all over the place. Rather than being consistent, they would be totally dependent on some other factor. You'd get wide swings in the amount of the heavier carbon in the specimens, depending on who knows what. But this could not be interpreted as something that was an autotroph. It couldn't be getting its carbon out of the atmosphere, because you wouldn't get that range of variation in the isotopes. So that shows that this thing was a heterotroph. It was getting its carbon from a range of sources which have themselves different isotopic ratios. So it wasn't even a specialist heterotroph feeding on a particular type of plant or something like that. This looks like it was just breaking down existing organic matter from a wide range of sources, whether that be bacterial mats or pre-existing land plants or breaking down, or even animal remains, who knows. But it was a wide range of sources, which means it was a heterotroph, which means fungi stands up to scrutiny. So this was interpreted by Boyce et al. As some sort of giant fungus. There are some alternatives that have been suggested during this sort of time interval from 2000, but basically the consensus has been going towards it's either a fungus, a weird one which has evolved a structure that we don't see in any other fungi, or it's something completely new. Some totally different form of life that evolves some sort of multicellular structure, and yeah, you can imagine, as tempting as that is, you need really strong evidence to try to pin it down to that type of conclusion. Because to conclude that there was another branch of life that evolved not just multicellularity but gigantic size, that means that we've missed something really quite significant, and it's a major discovery. So you need really strong evidence to go down that route. So, on to 2026. Right, the new paper is by Corentin Laurent et al. Sandy Hetherington is the corresponding author. A long list of authors in the middle, because there's been quite a lot of analysis went into this. and it's published in Science Advances, which means it's open access. And what we have here is a series of analyses based on effectively one new specimen from the Rhynie chert of Aberdeenshire in Scotland. If you've not heard of the Rhynie chert, it's an extraordinary deposit. You can't find any outcrop, it's all subsurface underneath agricultural fields, but you get lumps of it coming up when it's ploughed and so on. And it is basically hot spring deposits from the early Devonian that preserve exquisite detail of not just early land plants that lived along the edges of the springs, but also animals that lived amongst them as well. So the first harvestmen, the first springtails and insects. There's all sorts of wonderful things in this site. I'll have to do a whole video on the Rhynie chert one day because it really deserves it. But it's most famous for the land plants and the cellular structures that it preserves inside them. And they do occasionally get Prototaxites there. And this new paper is based on the largest known specimen of Prototaxites from the Rhynie. By other standards, it's quite small because you're looking at, loose blocks, something like this, and so you're looking at specimens a few centimetres across. But it's beautifully preserved, and the fine detail in here is remarkable. There's a lot of organic material in it, and so you can do lots of analyses of that organic material in a way that you might not be able to do quite so well in other samples. You can also use all sorts of quite advanced microscopy techniques to look at the fine structures of these things, which they've done as well, and found that these tubes are really quite complex and peculiar structures themselves. Many of them are banded. They actually have annulations around them. Many of them are very irregular in their diameter, even on very small scales. They sort of branch into little fibrils and filaments, dividing into smaller and smaller branches at the end of some areas. In fact, one of the most interesting things that they discovered from this side of it is by using lots of quite complex stacking and confocal type microscopy, they've shown that all these very fine branches, or fine tubes, are actually connected to the large ones. And they separate at these sort of bright medullary spots, where these coarse branches suddenly divide into lots of fine ones that then form the fine tubular meshwork. It's a really complex and interesting structure which doesn't really match up with what you would expect from fungi at all. The main bulk of the results, though, comes from chemical analysis. And in this case, what they're looking at is not necessarily the molecules themselves, but absorption spectra. So they're using FTIR microscopy or spectroscopy to look at the absorption bands of the various molecules that are present in the specimens. And that tells you potentially which molecules are there, because each type of molecule has a slightly different spectrum of absorption bands. But more importantly, in this case, it allows you to compare directly with other organisms in the same deposit. And in the Rhynie chert, there are rather a lot of fungi, and plants, and lichens. So even without knowing exactly which compound is responsible for these absorption lines, you can look directly at this wide range of things in the same rocks and say, okay, is this the same as what we see in fungi? Does it have a completely different spectrum? Does it look like it is just a slightly modified version? And you can plot these spectral lines on, in this case they've used a principal components analysis, which is, it's a complex way of analysing very diverse data and drawing out similarities and differences effectively. Could go into it in quite a lot more detail, but it actually removes a lot of the signal of exactly what it is that you're looking at as the differences, but it does highlight differences and similarities. It's a very widely used technique in paleontology and other fields, so no problem using it. But what you end up with as a result here is luckily something very, very clear-cut. it's totally different. Compare the fungi and the animals and the plants, and the absorption lines from Prototaxites falls into a totally different region of the plot, all on its own. It doesn't overlap with other groups, it's just completely different. So this kind of scuppers the whole view of them being fungi, because if they were fungi, then even if they were fungi which had evolved some other sort of organic compound which formed the bulk of these structures in Prototaxites, you would still expect other organic components to be the same, and therefore you would get overlap with something like the fungal cloud. And it's just not there. So this in itself is pretty strong evidence that you're looking at something very distinct from fungi. To test this further, though, they also included biomarker analysis, because fungi produce various types of molecules that can fossilise, or at least relics of them, can fossilise and be recognisable. And so they also took samples from the matrix and from the Prototaxites and looked for fungal biomarkers in these samples. Specifically, they looked for a thing called perylene, which is known from the Rhynie chert itself as a biomarker of a particular type of phytopathogenic fungi, ascomycetes in particular, so things which attack plants. And these were already known to have been preserved in the Rhynie chert, within fungi of this type. So they took a sample from the sort of PT matrix, and they took a sample from the Prototaxites. No sign of it in the Prototaxites, but it was there in the matrix. So again, it's just showing that if it is a fungus, it's not any sort of fungus that has that type of biomarker compound within it. Okay, so what this leaves us with is basically no evidence whatsoever that this thing was a fungus. And it's not just that they failed to find evidence, they failed to find evidence that should have been there if it was a fungus. They also found more detail of the microstructural morphology of the thing, which also doesn't match what you see in any known fungi. So, could this potentially still be a fungus? You can't categorically 100% rule out that it is some extremely aberrant fungus, maybe derived from one of the more obscure groups that we don't really know much about, and has evolved an entirely unique architectural structure and has a different spectroscopic signature to any of the other fungi preserved in the Rhynie chert. Yeah, I mean, you could argue that it was still possible. But what they've shown, and I think pretty convincingly, is that that is now actually very unlikely. So if we assume for the moment, and this may change if someone finds another way of analysing it and proves that there is some sort of fungal signature there, but if we assume for the moment that they're right, this isn't fungal at all, then what on earth is it? Because you're left with not that many options now. And in fact, the authors do quite a nice little summary of the various possibilities for a large macroscopic multicellular organism based on all of the ones that we know about. Because multicellular organisms are basically divided into a few major groups. So you've got opisthecons, which is the group that includes animals and their relatives, but it's basically animals and fungi fall into this group. We've got archaeoplastids, which are effectively red algae, green algae and land plants, all of which have independently evolved macroscopic multicellular body forms. And then there's also the stramenopiles with brown algae. So multicellularity and large size is obviously something that isn't restricted to animals. It's something that can evolve in multiple groups independently. And what the authors have done here is summarise the various groups in which it might be possible and ruled out basically everything. So let's have a quick run through them. It's quite instructive. So prokaryotes. So these are things like bacteria and archaea. You can rule out because it's morphologically complex. There's nothing, even amongst the sort of pseudo-colonial biofilms and things like that, that you end up with these some sort of bacteria. You don't get anything like this internal complexity and the ability to create large structures. So things like the tubes I'm thinking of here, they can't cooperate in that way. So can rule out prokaryotes entirely. So you're left with eukaryotes. These are the things with the complex cells that includes plants, animals, and most other things that are familiar. Oomycetes. Again, complex multicellularity is something that is not known in any oomycete. If it evolved independently in this group, then okay, maybe you'd be looking at something being possible there, but at the moment there's nothing that is even vaguely similar. Cell walls is a way of distinguishing it from animals. These things have very thick cell walls. Animals have cells, but they don't have reinforced cell walls. So seeing that is more of a feature of plants, but it rules out animals. This thing wasn't an animal. Straminopiles, so the brown algae and things like that. This tube-like morphology, the structures within it, is totally incompatible with anything that's ever been seen in a stramonopile. So we can rule out that on that basis. The red and green algae, well, they are entirely aquatic. So just being terrestrial is a really big feature for ruling out those. You could argue that they adapted to terrestrial ecosystems, but that's a huge step. And if they had managed to adapt to terrestrial ecosystems in any form, and then had evolved enough to create these gigantic, monstrous columns on the land, you'd think we'd find some other evidence of that in some of these deposits. And there is no sign of red and green algae ever having managed to overcome the difficulties in becoming terrestrial. So again, they've ruled out that one. Land plants, nah, it's not that, because it's a heterotroph. This goes back to the isotopic data from the previous work. This is something that is obtaining its carbon not directly from the atmosphere, but from some pre-existing organic matter. And that also, of course, rules out the red and green algae and the brown algae, which are also autotrophs. So the fact that it's a heterotroph rules out most things anyway. Lichens is another potential possibility, but there is no evidence in any of this data for photobionts, things like other algal components of the structure, because a lichen is a symbiosis between algae and fungi, effectively. It actually gets a lot more complex nowadays, apparently, but that's how they're traditionally seen. And there's no evidence for any of these components. And there's also no compositional heterogeneity, so you don't see different parts of it appear to be made of different substances, which you'd expect to see in a lichen or in any other really integrated symbiotic organism. So that also doesn't make sense. And then fungi. This is where the paper is getting really novel, because it is ruling out fungi by the complex branching pattern, the medullary spots, these little clear patches, by the lack of the fungal biomarkers, and also from the FTIR data again, which shows that biochemically this thing appears to have been entirely distinct from all of the fungi that are known in the same deposit, preserved in the same way. So their conclusion from that is most likely that there is no affinity with a known complex multicellular eukaryote, with any of them. So that means that this is, if they're correct, a different branch of life entirely that has evolved a complex multicellular and terrestrial lifestyle. That's getting quite intriguing. And that is as far as the authors go, because they just don't have any further information as to what it could be, or at least they don't have anything they're telling us. But the conclusion is that it's something unknown. Is it possible for us to go beyond that and start thinking about which groups of organisms may conceivably have evolved this sort of structure? Kind of, but it verges really into speculation at that point. So you can look at other groups of protists, for example, that might have some hint of a similar structure. And there is a group called the spongomonads in the sarcozoans, which are basically things like rhipidodendron, which actually have tubes within their sort of almost frondose-like structure. So you've got something with tubes in. Does that mean that it's plausible as a precursor to something like this? We just can't tell, because the problem is that it is such a derived state that we're seeing this set of tubular structures in, that as far as we can tell, there's no real similarity. And tubes are things that have evolved in so many different groups. And could it be an entire group of eukaryotes that we don't have even small representatives of anymore? That's probably just as likely as it is that it's a group that we know already, but haven't got anything that evolved to form these sort of sizes or structures. It's just a huge, great unknown. And this is sometimes really important to recognise in science, that it's very tempting to say, well, we know about all the rest of life, let's just sort of look at it all and see which one matches. And sometimes you get a structure like Prototaxites that is so highly modified from anything that might be related to it that's alive today, that there is no obvious link. We can't look at what is already known and say that we think we know what it is. And this is potentially a really important lesson for us, because we've been getting society as a whole, I suspect, into a state where we think we know most of the big picture stuff nowadays about how life has evolved down its various branches. And being woken up with an organism that appears to be something entirely distinct from everything we understand is a really good wake-up call. We do not know everything. We can now see that there are structures and organisms that evolve down a completely different route to anything that we have seen before. We don't know anything about how these things reproduced. There are all sorts of peculiarities, like they appear to have grown in sort of phases. So they stick as one lump for a while, and then there's a rapid accretion of another layer, and then you get a layer there with sort of plant material being incorporated into it, and then you get another layer on top of that. It's a very different sort of growth structure to what we're used to seeing as well. And so this is telling us that this thing that we've been trying to squeeze into first plants and then algae and then fungi, we shouldn't have been doing that necessarily. If it looks completely different to anything alive today, maybe it was completely different to anything alive today. Now, yes, there's a bit of a subtext there for those who have been watching a lot of my videos. You'll know that I'm also sceptical of things like the Ediacaran biota. Many of these weird Precambrian things being squeezed into early animal evolution, where to me they really don't fit, but it feels a bit like desperation trying to fit these things into groups of organisms that don't really look anything like them. And I've long thought that more likely explanation is that these fronds and articulatans, things like the Dickinsonia and Charnia and so on, could be an independent origin of multicellularity from perhaps a stem group animal, perhaps something more distant. But a different group of life, a different branch of the evolutionary tree, which we don't have a modern comparison for. And this thing with Prototaxites is really just, again, a reminder that we shouldn't be too arrogant in thinking that we do know everything and that we can just sort of squeeze things in and cram them into spaces in the evolutionary tree that we don't know much about yet, because they've got to go there somewhere. It's just really interesting. It's a new perspective on a different organism. We are hopefully going to someday get closer to it. We can start looking at things like if there are any peculiar biomarkers that are being preserved inside Prototaxites, and start looking for that amongst all the swathes of protists, for example, that are out there. It may be we never find a match. It may be it always remains a mysterious branch of the tree that we can't pin down. But on the other hand, maybe one day we will be able to find a little bit of chemical evidence or even structural evidence that supports a close association with something tiny and obscure, which today doesn't manage anything like the grandeur of its previous existence. We'll see one day, but it's a really nice one. Thanks to Igloo again for pointing it out. I knew this one was coming because I think there's been a preprint up for a while as well, and I've seen talks about it. But this is another really nice news story. Hope you enjoyed that, and we'll be back with probably some new fossils from around here I've got coming up next. Found a new local fossil site, and it's quite interesting. See you anon.